All right, so as I was saying, the electron moves from point A to point B. Non-cyclic, doesn't cycle back on itself. The electron enters from the water in the second, or photosystem 2, not the second one, photosystem 2, through the pump, gets excited again energy-wise in photosystem 1, passed to NADP plus reductase, dumped onto NADP plus, filtered over or shuttled over to the Calvin cycle and deposited right there, right? And then that's food, and you eat that, and that's where those electrons are that you use for in the mitochondria for respiration. All right, so that's non-cyclic photophosphorylation. What happens if we get to a situation where we're running out of ATP? We need to make more. Well, plants are very clever. They can engage in something called cyclic. This time I've added that gray arrow right there. Look what happens. The electron, when it gets excited in energy the second time, instead of going to NADP plus reductase, because we have enough of this, you cycle it back to the PQ and cytochrome complex, back to this pump, so you pump more protons and make more ATP. So depending on the cell's needs, an electron could be in the cycle a couple of times. Now, eventually all electrons exit through the non-cyclic route. Remember I said electrons, you can't strip another electron off of water until the electron is exited. So while that electron might go around that cycle a few times to build up ATP, it is eventually going to have to find itself on NADP+. Right? Plants will switch back and forth between cyclic and non-cyclic, depending again on their ATP. All right, back to that picture we saw at the beginning. I want to focus now on the stomata. Notice what's happening in the stomata. The stomata is how the plants are exchanging gases. This is how the carbon dioxide gets in for the Calvin-Benson cycle or the dark reactions, and this is how the oxygen gets out from the from when we split water during the light reactions. Okay, and that's all well and good and normal the way it's supposed to work. But as I said, there are guard cells on the stomata, and they can sometimes close. So you might be thinking, well, why would they close? Is there a time when plants? I mean, it's kind of like saying, spend half a day, stop breathing. No, exchange of gases, that's not going to work. So the reason why the stomata might close, though, is while the stomata do the good thing of exchange the gases, water, dehydration can also occur through stomata. Not a big deal with plants that live in areas in the world in which, the weather's temperate. Rains and it doesn't rain, it rains, it doesn't rain. In other words, it's not like a desert, or not like, or what we call an arid region. Deserts are arid regions. Or like the plains are known as semi-arid, which means it rains, but it's more on the dry side. And you know that farmers out in the Midwest, in the middle of a drought, they can obviously lose a lot of their crops. Now, plants live in all these areas. They live in the ones that aren't arid. They live in the desert, right? Cactus and that sort of stuff. And there are plenty of plants that survive in semi-arid regions. So there's this question. How do these plants that live in these really dry climates, how do they deal with opening their stomata, which is necessary, and yet how do they deal with the fact that they lose water through the stomata when they're open? Okay, well, when it gets dry outside, what they do is they close their stomata. Boom, problem solved, right? Carbon dioxide that's coming in through the stomata, the plants will store a lot of carbon dioxide. So inside of the plant's tissue, there'll be plenty of carbon dioxide for a while. Now, can the plant keep its stomata closed forever? Well, no, of course not. Can't forever. It's like holding your breath. You can't hold your breath forever. But nonetheless, when the stomata close, there's still plenty of CO2 to carry out effective photosynthesis. It really isn't the lack of CO2 that, well, it is kind of, but it's more the accumulation of oxygen that becomes the problem. You see, when the stomata are closed, there's nowhere for the O2 to go. So oxygen levels begin to build up inside the tissue of a plant. You think, well, that's not a bad thing, right? Plants need oxygen. Well, they do. But the problem is it becomes an excess. And there's a problem when it becomes an excess because of the enzyme Rubisco. This one here. Now, you remember back from our discussion on the enzymes, what gets an enzyme and a substrate to come together, right? They fit. So they have an affinity for one another. Rubisco has an affinity for carbon dioxide. Of course it does. It has an affinity for carbon dioxide and RUBP so that it can create this first